vaccines as a part of being considerate to others, not only to protect yourself but to protect everybody else around.
Sir, am I audible?
Yes, yes.
Sorry for the delay, everyone. Good evening, one and all. I would like to welcome you all to day 2 of ATAL sponsored 2-week FDP on computational biology in therapeutics and theranostics 2022, organized
We do every week.
Allahabad. Today, we have with us two very eminent speakers, that is Dr. Rakesh Mishra sir from TIGS Bengaluru and Dr. Krishna Mishra ma'am from IIIT Allahabad.
We do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP. What is the situation and where is the red alert or where is the cases are going down and so on, and they respond. This is the overall city average
Perspective. This session will be recorded as per AICTE guidelines, and if any of you have any queries in between the session, I would request you all to please put it in the chat box. Now, before starting the session, I would like to invite Dr. Sindhu Kumar Samanta sir for introducing the speakers.
Thank you, Ananda. So today among us, we have
we do every week.
Misra, who is one of the renowned scientists in India. He was the 6th director of CSIR, Center for Cellular and Molecular Biology, that is CCMB Hyderabad, till April 30.
We do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP. What is the situation and where is the red alert or where is the cases are going down and so on, and they respond. This is the overall city average.
Professor Mishra worked as a postdoctoral fellow at MBU, Indian Institute of Science, Bangalore, during 1986 to 1988, and he worked in the area of non-BDNA conformations and DNA topology. Then he moved to CCMB Hyderabad and worked as a scientist and studied transcription initiation in prokaryotes, chemical recognition, and cleavage of DNA during 1988 to 1992. During 1996, he worked at the University of Bordeaux, France, on antisense oligonucleotide mediated knockdown against protozoan parasite, and further he investigated small nuclear RNAs in Xenophas oocyte.
We do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP. What is the situation and where is the red alert or where is the cases are going down and so on, and they respond. This is the overall city average.
As a scientist at CCMB Hyderabad in the year of 2001 and became the director in the year of 2016. During his stay in CCMB, he worked in the various areas of biochemistry and molecular biology, mainly comparative and functional genomics of non-coding DNA, organization and regulation of Hox complexes in animals, epigenetic regulation and development. He has got more than 95 articles published in national and international journal of high repute.
We do every week.
It's important to mention here that the ATAL Incubation Center was established at CCMB under his leadership, which provides life sciences entrepreneurs and infrastructure and mentorship needed to encourage innovation.
We do every week.
In addition to this, he is an elected fellow of J.C. Bose Fellowship, Indian Academy of Sciences, National Academy of Sciences, India, Indian National Academy of Sciences, Andhra Pradesh Academy of Sciences. With this brief introduction, I would like to invite Professor Rakesh Mishra sir to deliver his talk on genome-based environmental surveillance of infectious disease, an emerging and essential tool for public health system. Over to you, sir.
We do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP. What is the situation and where is the red alert or where is the cases are going down and so on, and they respond. This is the overall city average, but this is the different STPs which also indicate the wards from which part of the city cases are going up and down. So I'm just putting it like to note. It's really an honor to speak before her in this meeting, and it's really impressive and encouraging for all of us how she has been pushing the quality science in very demanding conditions and very limited resources. So with that, and so it has been we do every week real I would say extraordinary opportunity. We do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, and they respond. Is this is the overall city average, but this is the different STPs which also indicate the wards from which part of the city cases are going up and down. So you continue in Tata Institute for Genetics and Society where I'm currently working. So with the we all are just recovering from the pandemic which has shaken the world, where 62 crore cases till now and 65 lakh deaths reported. And US alone, we do every week crore cases and 10 lakh deaths. Remember, US population is, we do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, and they respond. Is this is the overall city average, but this is the different STPs which also indicate the wards from which part of the city cases are going up and down. So you know, more deaths in the country. It's very difficult to actually count. Besides that, there has been enormous economic disruption, probably many times we sometimes miss. In the world, according to expert estimates, $16 trillion economic disruption has taken place. And another factor which we do every week should get is the psychological impact on the younger population who have not gone to say. We do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on. But this pandemic has changed the research and healthcare approaches probably forever. And the reason for that is that, which will become obvious, that it has catalyzed a research revolution to understand the virus. In 2 years, 2.5, 3 years, what we have understood about a disease is something enormous. And a novel virus is discovered and a vaccine is made within a year of the discovery of the way we do every week, unheard of, which never has happened in the history of humankind, that a vaccine is available, and such good vaccine, and dozens of vaccines are available against this virus, which has helped us fight this pandemic. And testing and surveillance that we do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, to isolate RNA, make DNA out of it, and estimate how much of RNA was there and so on. It's quite challenging job. And what has also emerged during this experience is the genome surveillance, which is an extraordinary tool, which is to understand the evolution of the virus, to understand the infectivity or immune escape features in the virus, and to trace the path of the spread of the virus, which was not used. We do every week ever. Since we are going to focus on this aspect, I'm going to talk about what is genomic surveillance. So this is a process of constantly monitoring the pathogens and analyzing their genetic similarities and differences. And why this is important? Because we do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, and they respond. Is this is the overall city average, but this is the different STPs which also indicate the wards from which part of the city cases are going up and down. So you don't have to worry about evolution of the infectious disease agents, alerts the spread of the pathogen, and develop countermeasures like vaccines. why so much of variants, so many mutations in this virus? So, in fact, the mutation is the main strategy of many pathogens and particularly the viruses. And coronaviruses in particular, they are very fond of mutations because that's their only weapon they have to evade. We do every week and keep infecting and improving their weapon. We do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, and they respond. Is this is the overall city average, but this is the different STPs which also indicate the wards from which part of the city cases are going up and down. So you don't have to worry about each, and we may not even notice a mutation. And let me tell that more mutation doesn't mean virus is more dangerous. It is just a matter of chance. And this virus has been accumulating mutations at the rate of 2 mutations per month. So that's what we have understood. And before I go into the what is the surveillance, genomic surveillance means, this is an interesting figure to show the lines that you see. They are the mutation rate and different parts of genome. We do every week a lot of mutation in this part of the virus, which is the spike gene. We do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, and they respond. Is this is the overall city average, but this is the different STPs which also indicate the wards from which part of the city cases are going up and down. So you don't have to worry about each one the virus. So this war continuously goes on, whether against drug or against vaccine or against the naturally evolving immunities. So the variant genomic surveillance can be of two kinds. One is the clinical samples and the environmental samples. And I'll give you some context of how this we do every week are decided. So, so strategy for genomic surveillance is, of course, sequencing as many samples as possible. So number of sequences we do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, and they respond. Is this is the overall city average, but this is the different STPs. You see immune escape, you see vaccine breakthrough, all those things, and mortality and worse symptoms are not of any advantages to virus. It's just our bad luck. But the selection of the virus is based on vaccine breakthrough, immune escape, and higher infectivity. These are the selection criteria for the virus. And we should sequence enough that we get we do every week these aspects. Now, the strategy for this surveillance is, is the following. So, for example, when you want to collect a sample, what kind of samples you want to collect? So, one is unusual clinical symptoms. So we see people are more. Say we do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP. Is other infections or immune problem, or it's new variant that has come which is causing this versus clinical symptoms. Then vaccine breakthrough cases are important to sequence because we can ask whether the vaccine is fading or not working, or it's a new variant which is evading the vaccine. Similarly, reinfection, whether it's immune deficiency or fading of the immunity, natural immunity of the virus, of the individual, we do every week come which is evading the immune system. Similarly, local super spreader incident, when see a lot of people getting infected, it is important to sequence there to find out, is it a human behavior? The lots of people had a party together in a closed hall or something, or a new variant has come which is now very efficient and so on. And then, of course, in addition to this, one should keep doing geographically randomized samples to keep the track of the trend and the new variants of the virus. So that we do every week how you select the samples. And more or less, this is the kind of thing that is happening. And at CCMB, we have a Gear 19 tool. You can go and play with this. It shows what are the sequences. This is a few months old slide. How many sequences have been done, how many lineages. These, let's say, 51,000 mutations have been cataloged, and these are various variants that have appeared. You can see the way we do every week here that these are different colors are different variants which are listed. So they emerge, they go down, some new emerge thing comes and goes down, another one, yellow one comes, and then the pink one comes, and so on. So this is where the pink one is when it was BA.1 and BA.2. So this way we can keep track of, these are the individual samples that we take, which when the throat swabs, we get the sample. Now we do every week, we are going to talk about environmental samples. So we don't go to any individual. You just pick the samples from either wastewater or air or zoonotic samples and estimate the load of the virus from there and ask what is the burden of the virus. So this is an unbiased and early warning way, which I will explain to you now, and it's convenient and cheap because you don't have to find people and collect samples. We do every week from the pond or the STPs or air from wherever you are sampling. And I just want to say that it is also relevant beyond SARS-CoV-2. SARS-CoV-2 is probably a platform to launch such strategies for public health and monitoring of infectious diseases. So here is the first example I'm going to give you. This study which we started a couple of, we do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, and they respond. Is this is the overall city average, but this is the different STPs which also indicate the ward. So these are the places where we were arranging samples to NIRI from different STPs and measuring the load of virus in the wastewater in these cities. We do every week. The wastewater surveillance we have done based on STPs, based on open drain, because that was done deliberately because many cities don't have a fully everything is in STP manner, their main drains, and many cities have only drains and so on. And we have also monitored the lakes near Hyderabad, which have where population samples are put or some are totally in the disconnected to the population. And this we do every week, so that at a given time, like this is in the July August period of 2020, when we estimated the level of the SAM infection load of 1,27,000. At that time, based on the city numbers, it was about 20 times more than what was being reported. So this number turned out to be the correct one, because later when we did the sero surveillance, we do every week during this period, it shows the infectivity rate which matched this number. So which means the city numbers or the patient samples will depend on first proper reporting is happening or not, whether we are doing enough testing or not, and so on. The kit is working and so on. Here you go and take the sample from an STP. So here we have used 9 of them. We know the total capacity of these 300, 603. We do every week uh, uh, liters, and then the time duration and so on, and we can calculate this number. So actually you can, without going to any house, without going touching any person, you can tell how many people are infected in the city. So that was from STP. This is now we are currently till now we are doing 20 locations, which is showing in this map. These are open drainage systems in the Hyderabad. So we do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, so that even the open drainage we can have a fairly accurate idea about this is in the overall case load, and this is the RNA copy number per ml. You can calculate from this. You can also calculate from the RT-PCR or from the sequence. We do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, and they respond. Is this is the overall city average, but this is the different STPs which also indicate the wards from which part of the city cases are going up and down. So you don't have to worry about each graph. So this means not only the case load, but even the variants we can detect we do every week. Here is the dynamics of the case load in these 20 different locations, which is listed here. You can see that as the time goes, the cases go up and down, and so on. So you can follow in different catchment area the case load that is happening. And I will give you an example that this thing is from Allahabad, your program. So we have done at this time samples from Allahabad based on wastewater surveillance, and this is the based on the say we do every week from the numbers case reported. You can see the trend is very similar. This is the same timeline here, same timeline here. You can see that peaks or the where there is sharp bend in both the wastewater or the case reported is very similar trend. So which means with the wastewater, actually you are getting the real trend that is happening, which is revealed by the cases reported. We do every week testing. And this is the trend of the cases over different period of time. Let's say STPs or open days in Mumbai, Nagpur, Maharashtra, other cities, Delhi, Chennai, Kolkata, and so on. And you can see that somewhere there are cases going up and down during the same period and so on. So as we know that India is such a diverse country, there are various pockets where the cases may be going up and down, and therefore we do every week surveillance give very good view of what is happening out there. So that was the story of Hyderabad and a few other cities. And since we began in this analysis about a year ago in Bangalore, where we had tremendous cooperation from the Bangalore Municipal Corporation and wastewater organizations who were sharing is the sample, and these are the 28 STPs of we do every week. Bangalore city is one of the largest STP in the world, maybe after the Brazilian cities, and it covers 80% of the population. So what we are going to talk about is about 80% of the Bangalore city's situation, and this is very nicely funded by Rockefeller Foundation, and several organizations have been involved to conduct such studies like NCBS, the institute in the campus where we are, and we work together uh, for doing these surveillance. We do every week. We have plotted the red is the COVID cases as reported from the city sampling, and the blue is the trend from the wastewater. You can see there are certain times where we are seeing the peaks which is not seen in the reported cases because you don't test enough. For example, this peak is during the Sahara period last year, so which means the Sahara people had more activity and cases have increased. And this is the third wave, and this is the after the third wave more recent thing, and you see the cases we do every week. We see the actual numbers here. The trend is very similar. Some of these things are missed because we don't test enough, but these cases not only give the trend, but also depending upon the time lag of 7 days or time lag of 4 days, you get the alert 14 days in advance or 8 days in advance. So from there now we have made models based on which, so we, by the way, we do every week testing of all the 28 STPs in Bangalore, and we give the number to BBMP, what is the situation and where is the red alert or where is the cases are going down and so on, and they respond. Is this is the overall city average, but this is the different STPs which also indicate the wards from which part of the city cases are going up and down. So you don't have to worry about each graph and all. It's just to tell you that extraordinarily granular and scientifically robust data is generated using this without touching one person. So we can tell that in this locality cases are going up, they are going down, and so then city authorities can go and take care of what is happening there. Too many activities, or new cases or new variants are erupting there, because all this is also done sequencing. So we know where a new variant has come. That's why cases are increasing, or say human activity that is leading to this increase. So you can imagine the immediate use of this kind of information. And as I said, sequencing gives very good view of, you can say different color means different variants. As you see, large number of variants are seen in the wastewater samples, which is not seen actually in the samples collected from the individual, because that is very small fraction of the actual population of infected people, while the wastewater is a full net sampling, and you get the full picture of the virus variant. So this is again similarly you see different color is different variant. You see lot more variant than actually you see this is the clinical samples where you see very few because the numbers are very small there and some variants are missed. So therefore this variant calling also is more efficient in the wastewater samples. Now, if you notice in the previous things, there are a lot of cases where we say unassigned, and you can see a lot of variants are unassigned. Here also, there are a lot of variants are unassigned. So, what is this unassigned? With so much of sequence information, computation, why we still are not able to assign? Is it a sequencing error? Or this is a bioinformatics limitation or error, or is there some unmappable but still SARS-CoV-2 recombination that has happened because that will not be easily matching with any variant. So we asked the question whether because that was a big problem, whether this virus can recombine like common cold virus. So is this recombination new trick of the virus? If so, how successful it has been, and does it mean more deadly prospects ahead of us? And as you can see that unassigned cases during the peak, they are very high in number. So what we did was we collected the data from the publicly deposited data from India and asked if this recombination is a new trick of the virus, and if so, how successful it is. So the way we ascertained is was that we asked what is the recombinant parent. So this is one parent, this is another parent node. These two variants have to be in the same time, same city or same region, then only they can recombine because they have to be in the same individual. So we see that let's say one parent node is in Jammu and Kashmir was in Delhi, close enough, then MP, Tamil Nadu, so on, and they are from the data within 1 month period, 12 days period, some are coming from the same day and so on. So which means these are indeed variants, recombinants of two variants combining together. So, of course, it has been there, but these are all not flourishing very much because they come and go. So, but we cannot write off the recombinant ability of this virus, and the virus has more tricks in its bag. And this is to show you that various different variants, how they are there during, let's say, January to July period. So you can say BA2, but there are lots of this XM or XQ. These are actually variants. So they are there, but not very impressive in the number. So what we learned from this is that wastewater surveillance is effective method for genomic surveillance or even normal surveillance, and it is essential component of the patient sample surveillance because it gives us the full picture. And we have been able to convince the municipal authorities that the wastewater surveillance is effective method for genomic surveillance that complement the patient data. And it can be used to measure the infectious diseases, load and trend in the city, and extremely useful for the public healthcare system. In next 2 years, we are going to continue this, which is this program, which is funded by Rockefeller Foundation, to focus on other infectious diseases, including antimicrobial resistance, which is a major problem that we can see forthcoming. The zoonotic surveillance under the One Health program, and the sustainable system to develop so that every city can have this. So we want to make a robust protocol, train people, indigenize the reagents that are required so that in few thousand rupees you can assess the infectious disease load in the city, and by the trend you can see what is coming and going and take appropriate actions. So that is very important for the healthcare authorities. This is lessons learned and way forward. So from our study, even the World Health Organization has made a environmental surveillance of SARS-CoV-2 a complement to public health service surveillance guideline, where they have referred to our work, which shows that this is a noticed and very important way of handling this and maybe future pandemics. So sewage surveillance gives a quantitative and qualitative both estimate. It can be used as a reliable and unbiased surveillance method. Uh, it's also an indispensable tool for early and easier detection. Very easily, 1 week before you know whether the peak is coming or not infection, and you start preparing and don't run out of medical necessities. And certain efficacies and strategies can be put in place And along with SARS-CoV-2, several infectious agents and biomarkers associated with disease can be detected in wastewater surveillance, which we are standardizing for common diseases, infectious diseases in India. And surveillance can also help in understanding the antimicrobial resistance emerging in the microorganism. And we are having really interesting insight from the studies that are result that we are finding. Now, next 5-8 minutes, I will talk about yet another way of environmental surveillance, which is surveillance through the air. Initially, the contact and droplet mediated transmissions were considered as the major modes for transmission of SARS-CoV-2. The alarmingly increased number of cases of COVID-19 globally raised the possibility of airborne transmission, which WHO denied for quite some time. And consequently, the CDC and WHO later discussed the possibility of airborne transmission of SARS-CoV-2. And airborne transmission has been previously reported for SARS-CoV-1 and MERS-CoV. So we decided to, this is very early in the, just around the, even the first peak, we started planning and implementing the strategy for air surveillance. So here we collect the air sample using a little, it's some kind of a little vacuum cleaner, which has a gelatin membrane as a filter. So air passes through it and a virus gets stuck in the gelatin membrane with multiple layers. And then so that's a process of air sampler with a flow rate of 50 liter per minute. And then we take the gelatin filter out and extract the virus from there, and then do the RT-PCR and analyze the results if we are finding any virus in the filter. So that means in the air we can detect the virus by this method. So we planned this extensive study, taking samples from hospitals, hospital ward, COVID ward, ICUs, doffing area, OP corridors, many other places, mortuaries. In mortuary we had to do because there were lots of problems. People were thinking dead bodies are infectious and so on. So we established that nothing comes out of the dead body. So give respect to those. And then controlled condition, we had a specific rooms where we put infected people and then did the measurements as a scientific experiment. And we also collected samples from houses and of positive individuals, their bedroom, living room, bathroom, and so on. And conclusions were quite interesting, and some of that I will show you and summarize here. So this study was done in CCMB along with the IMTech in Chandigarh. So we had two places, Hyderabad and Mohali, and then we were sampling a number of hospitals and so on, and we did a lot of samples in 80 different locations and so on. And this, as I mentioned, in many places we did the sampling. And this is the design in the room where we have different distance, different time, keep infected and uninfected people, and when people leave, then also keep testing and how long the virus stays. So for this was a designed experiment for the COVID positive individuals, putting them in this defined space and checking 4 feet, 8 feet, 12 feet, what is the safe distance and so on. By the way, we detect the virus up to 20 feet distance, so it's there quite for quite some time. And throat swabs were collected as controls in these experiments. So here, I mean, I will just give two examples. One is in a family, two members were known to be COVID positive, and there were no room quarantine. They were freely using entire house, and then very soon, then all the 6 people, when they were the day we did air sampling, all the 6 people showed positives. Like they were all, you see the CT values are quite low. And when we did the air sampling in the individuals from 1.5 meter or 3 meter, bedroom, hall, lunch, table, bathroom, bedroom, everywhere you can see average is in a positive range of CT values. So that means the air was full of viruses in the entire apartment where they were living, because they were not quarantining, they were not using masks. And then we compared with another family where one person was COVID positive, he had symptom, and then COVID positive individual was then room quarantine within the same apartment, uh, room quarantine in separate apartment, and was shifted to hospital one day before the sampling. and the other family members were staying in the opposite apartment. And we went and checked this is the people, this is the infected individual, and two other individuals are negative. And we checked the patient room, there is a positive result, and family members bedroom, bathroom, hall and kitchen were all negative. So that means if you do quarantine and wear mask, actually you can prevent the spread of the virus. So summary of this study is that the SARS-CoV-2 is detected in the air in certain conditions. The chance of picking SARS-CoV-2 in air is directly related to the number of COVID positive cases in the room, their symptomatic status, and the duration of the exposure. The strategy of segregating hospital areas into COVID care and non-COVID care can help in controlling the spread in the hospitals, which is what hospital did, and it was useful. And the analysis, the air sample collected from household of the COVID-19 positive individuals suggests that if the patient is immediately isolated, after knowing that he or she is COVID positive, the cross infection to other family members can be prevented. And in closed and less ventilated rooms, virus can stay longer in the air. We checked that more than 2 hours. So when you use a public toilet, if somebody has used 2 hours before, you can still be infected. Therefore, always wear your mask. So based on this study, we issued a public advisory. This is now more than 1.5 years ago, and this study is now published also. So what was this precautionary measure based on such simple study was that avoid gathering in closed space. I'm saying this because this is still valid and we should all follow, and even normal time we should try to avoid so that we can fight better the infectious agents. Avoid spending more time in closed rooms where others are sharing in the office space and wherever. Keep the windows and doors open because cross ventilation is very useful. Allow the cross ventilation that reduces the infection chances. Wear mask, especially in the closed space, public toilets and committee rooms, even if there is no individual, because somebody may have been there 1 hour, 2 hours before, and the virus may be still hanging around. So this shows that by simple surveillance method and using genomic approaches, we can talk a lot about, tell a lot about the nature of the variants, nature of the virus, how to, by knowing simple things, how we can fight better pandemic of this stature, which I began by saying that how many crores people got infected and how many lakhs and lakhs people died. And if we have this kind of understanding, we can control these things better. So world is becoming smaller, and if a virus is detected in one country within a few weeks, it is everywhere in the whole planet. So that means we share the air that fast through travel and other means. So there will be debate now for quite some time, the origin of this virus, how it originated, lab made, all those kinds of things, though I don't subscribe to that theory. The variants, how they emerge, infectivity, symptoms, vaccine equity, a major issue. Although vaccine is there, but many people don't get it. And the world is one, so unless everybody is vaccinated, vaccination may have limited and only half the effect. Data transparency, very important. If you don't give proper data, then decisions and strategies that will be designed will be faulty. And infodemics challenges, you all must have heard all kinds of variety of rumors and so on, which is very unhelpful. Long COVID, mild variants, all those things, whether mild variant is good, I mean, that's not at all true, because mild variant will give one day strong variants and so on. So these the debates will go on. They have learned many things. We know what is right, what is wrong very often times, but lots of things like long COVID and still need to solved. But what we can do from all this study that I explained to you, we have to learn to live with this virus and the likes. Mask and vaccines as a part of being considerate to others, not only to protect yourself, but to protect everybody else around you and defeat the virus. Because if you don't get sick, but you spread the virus, this means you are helping the virus. So mask and vaccines prevent that. Avoid clustering in closed space and crowded. If it's not necessary, don't be there. And if it's necessary, wear the mask It's a small thing, but if we make it a habit, like we wear glasses, we wear topi, we wear other kinds of decorations. Mask also is something they are invading the culture in certain conditions just to mark respect for each other. And remember that virus spread only through people. It doesn't spread on its own. So unless we help virus knowingly, unknowingly, it will not go, not do anything to us. And this we know these are all the what is kind of origin of this virus, so we should respect wildlife, and more pandemics are in the line because we are invading the wildlife and exposing ourselves to the environment or to pathogen which we have never seen. So we have to learn how to respect wildlife. This planet belongs to all creatures. It doesn't belong only to human beings that we can do whatever we want. We have to see that all other creatures are in their places and we don't go and rob everything from a forest or wood and so on. And then preparedness for the pandemic in future at a global scale is work, which I think is, if $16 trillion is the loss of one pandemic, a fraction of that, if it is spent, we can probably prevent such pandemic or significantly reduce the impact of the pandemic in future. So with that, I would like to thank for the giving me the opportunity to talk about our work, which I think is of relevance to people and decision makers, policy makers, and so on. And a very special thank to BBMP and BWSSB. Availability of the sample, unfortunately, is a tricky situation. Many cities don't want to give samples because then they will get the true number, and they start to make a political criticism out of that. While BBMP has been very, very forthcoming and giving us samples, the Chief Secretary talking to us and taking steps which has helped them a lot. So in a way, we are setting really example to others to follow, because this is the way to do it. We must know what is happening. We must know the parasite load, the trend, and therefore and take steps to control them and not hide the facts and don't so if you close your eyes doesn't mean that then you for me to speak, and I'm trying to stop sharing so that we can I'll be happy to see that we could do so many things by collecting the samples. CCMB acted as a repository, validation center, testing center, training center, and so on. So that was a lot of opportunity to learn many things and contribute to whatever we could um, in our own way. So thank you very much for the opportunity to for me to speak, and I'm trying to stop sharing so that we can. I'll be happy to answer any questions or really happy to take the comments because lots of issues that I've discussed, they are quite debated, and some things we know, but some things we are still wondering what it could be. So we can all discuss and help each other understand. So thank you again, and I'm happy to take questions.
Dear sir, thank you so much for the wonderful speech. It was an honor to get to know so much from you. Now, I would like to ask everyone that if they have any queries, then you can please ask sir. And if you're not comfortable speaking, you can also put it in the chat box.
Questions in the chat. Agonia, please, there are written questions in the chat box. Please read them so that sir can answer.
Sure.
I can read one question, which is by
Okay, sir. There's a
Rajkumar.
question. Okay, thanks for the wonderful talk. Wastewater may contain detergent, which might denature macromolecular structure of virus. How the samples might have survived longer in the wastewater. What were the major sampling challenges?
So that is very important question. So we were measuring the fragmented pieces of RNA, and we are not measuring the virus. You are very right that in wastewater virus gets fragmented and broken, and there is actually no active virus there. But these RNA pieces, they are somehow preserved, especially in the with maybe complex with other kinds of fatty lipid or other kinds of components. So we collect everything and take out whatever is the soluble and diffusible and make a RNA preparation and remove everything else so that it becomes usable for molecular testing. So you're right, virus is broken, and what we are looking at the virus pieces, the RNA pieces, and but that is good enough to for us to calculate back, because each virus will have only one unique piece. So when we find n number of pieces of the same sequence, we know that those many number of virus particles were there. And since we know infected person how many virus particles they shed per day, so based on that we can calculate how many people were probably infected.
Similarly, another person has asked, Sir, I'm trying to understand how a single-stranded RNA virus is able to survive for so long in wastewater. Secondly, there are reports of human coronavirus 229E present in water samples prior to pandemic. So the result may be reflecting the sequence for the same.
I'm not sure about the before the pandemic who has found the virus in the water and what was the study. I'm unaware of that, but I can say your first part that which is very similar to the previous question, that uh, any virus, even the DNA viruses, will not be surviving that long in wastewater. So one thing is that we are collecting samples every day. So virus has to survive just few hours, because when these from the houses it reaches within few hours to the STP, and we have fixed time when the maximum data can be obtained. Collect the water and isolate the RNA from there. And as I mentioned, it is not the virus; it is the broken pieces of fragments of RNA of this virus particularly, because our tests are very specific, because you are using the primers which will read only this sequence. So this is fairly doable. Many countries are now doing, in some places you may have heard the polio virus is detected in some cities in US and in UK. Some of the probably are the vaccine parts, but some are probably there and they are mutating even. So wastewater, even if uh, uh, some in an area, if let's say 1400 people are living, even one person is infected, you will pick it. So that's sensitivity. So you can say that 0.1% of the infection level will be picked in the virus, and it's not for many days, it's one day, because next day the STP is emptied, so you cannot get any sample for the previous day. So these samples are as fresh as within 24 hours.
Okay, sir, I have one query. So this was wastewater you have collected, is it nearby any hospitals or any residential area as well?
So the STPs are the sewage treatment plants that
Ah,
are the whole city of that particular ward. So whatever in that ward is there, whether it's school or university or housing and so on. We haven't gone and specifically collected from hospitals, because many places don't have proper STP systems. And if you go and collect open drainage, then depending upon the time and your luck, you may find or not find something. But STP is the best where, and which will tell in that particular ward, if there are 1 lakh people living there, in out of 1 lakh, how many are infected, because all the water is going to that STP. So Bangalore has one of the best sewage system, as I said, one of the largest, and 80% of the population is covered. So it gives the full, almost full picture of the city. So maximum granularity we can do is ward wise, which is the contributing to the a particular STP. Beyond that we cannot do, because samples get mixed. But there are hospitals and there are housing complexes, and like for example, we are in now in Blisk Bangalore Life Science Cluster. So we are constantly monitoring our campus, lab premises and the hostel premises, and we know if somebody is infected or not from there. So you can do, so this can be designed based on your actually water, I mean sewage system of the city.
Okay, okay. So, sir, as you're saying, you are basically collecting the fragmented RNA, then followed by two artificial and from water samples. So, what
Hmm.
exactly you did in terms of from air collection? How do you collected
So
the
air
sample?
is that's a good question. So air is a different air we are taking from the air droplets or the micro droplets that are there. And in fact, the sample when we collected from the air, some of the after elution we infected in the cells and we found the infectivity in those samples. So in air you get very fresh because you are within 1 or 2 hours and those samples are still in some droplets and hanging. So they are infectious. So the air wastewater samples are not infectious, but air samples are infectious, and then give you the measure of in that air what kind of viruses and how much you have, and that they are infectious. So that is different. That is not largely not a broken one, but more intact pieces because they are actually the active virus particles.
Sir, as a water sample, you are saying, of course, you are using gene-specific primer. But still, sir, what do you think, depending on the water molecules, so sometimes that abundance of other organism, other viral infection, other bacterial pathogen infection, do you think it will interfere with your PCRs or findings?
Yeah, I understand your concern. And so when you take even the human samples, swabs, you have virus is something, but most of the DNA is actually human DNA in there. So PCR, that is the beauty of PCR and the sequencing reactions that if from the millions and millions of other molecules, you can pick one and identify specifically. So, like there is a question that whether we find some other bacterial and fungal co-infections. So there are two ways of doing this. A, you have to look for those. We don't know co-infection because co-infection will be done with the individual samples. In the wastewater, we don't know because that's a mix of everything. But we, as I mentioned, we are now looking for other infections like dengue, chikungunya, and other bacterial infection, tuberculosis, monkeypox. We never found monkeypox, so, but government, I mean, municipality wanted us to check, so we did that, and other fungal and things for so for which you are really looking, so use a specific primer. Other way of doing is that you do microbiome analysis, so the metagenome, what is called. So you can just do the sequencing of high throughput and then do the bioinformatics and uncover what are the various infectious region there. And that's how actually we are picking the AMRs and other new infections that are there in some quantities. So those studies are in progress. So, but that becomes then expensive because genome will be several thousand rupees, while just the RT-PCR to find one particular infection will be just few hundred rupees, even cheaper. So we can, so the our study is now every month we look for the full pathogen scenario, and every week we look for what we are actually following. In pandemic it is SARS-CoV-2. Some other time it may be something as malaria or whatever is requirement of that city.
No, this is a very wonderful work, a new thought. Sir, there's some more queries, sir. Did you find some other bacterial and fungal co-infection along with SARS-CoV-2?
So
Some
that's what I just answered, that co-infection I can't say from the wastewater or air sampling because you have to take some individual sample and then do the detection there. People have not seen actually anything, any major course of co-infection. Others have done that study. But as I said, some pathogens we are directly monitoring, and every month we are doing the epigenome, I mean metagenome. So looking at the full array of infectious agent that may be there. So we are doing that
sir,
and
another
continue.
participant, Parul, she has asked, any new method for air sampling?
That is a challenging thing because what we are doing is with the filter base. You can also, I mean, there are many chemists and biotechnologists here. You can actually pass the air through a solution, and where the virus will get trapped in the water from the air, and then you can collect that water after some time. So essentially, you pull the water bubble in the flask, and then let the air go, and so after some time take that water and go for testing. So that can be done. We haven't done, but that's a method. And some of you should use your entrepreneurial capabilities and design in new ways of sampling. So Parul asked this question, which is the relevance that the filters are expensive. They cost a few dollars, $5, $6 each. So that's expensive. So either we make our own filters and make them cheaper of 10 rupees, 20 rupees, then it becomes doable. Because imagine that you have to use many of them wherever you want to do, because you want to do certain numbers and so on. Or you use air bubbling system, so air is sucked and bubbled through the liquid in a conical flask with the air escape system, and then use that liquid. So this is possible, but we haven't used that.
Okay, sir, another person, Neetu Moria, has a question. What are the difference between airborne virus and waterborne virus?
So
What's the
by the
difference?
way, this virus is not waterborne. We detect in the water because it is stuck there and broken, and so we can just tell that how many people have contributed. So this is not a waterborne virus. A waterborne virus will be infectious when it is in the water and we drink that water. So this is not the case. Airborne is it is hanging in the air in micro droplets, and we breathe the air and it goes in our respiratory system and infects the epithelial cells. So that's the difference, is that we are getting one drinking and getting infectious through the intestine and stomach, another one is through the respiratory system because we are breathing that air.
Sir, another question from Monica Verma. Sir, as you said, we can identify particular pathogens through the PCR. So I want to ask whether you can use specific primer designed for that desired organism.
Yes, yes, absolutely. That's exactly what is done. So for if you want to say, let's say tuberculosis, then you have to use primers. You make the same DNA, RNA uh, uh, preparation, and then if you use primer for tuberculosis, if there is tuberculosis in your sample, you will amplify. If it is not there, you will not amplify. Or now people are making a variant specific test. So if you isolate RNA from your wastewater sample and use the primer, that will specifically amplify Omicron, or something will another variant will not be amplified, and something may be general. So this way you can distinguish not only the pathogen, different viruses are different pathogen, but even the different variants, as long as you know the sequence and design the primer appropriately. So it's quite possible, and it is exactly based on primers that you design.
Sir, one more question. In your opinion, what is India's preparedness for any future pandemic such as COVID-19?
This is very, very challenging thing, and my knowledge is limited. But one thing we know, I can give you some examples that when the pandemic started, the tests were costing 3,000 to 4,000 rupees, and there were hardly any availability, and there were lots of chaos. In within 1 year, we made a group, and with involved companies, our ATAL Incubation Center and the C Camp and some 50 companies we involved, and through proper funding, we could indigenize all the components. We tested each components, and we can now produce several million test reactions per day in the country at the cost of 69 rupees per test. And now we have done further improvement in this process with the help of many entrepreneurs, and cost per test material cost will be less than 20 rupees. So imagine where is 4,000 and where is 20 rupees. So this means, and also remember that at the peak of maybe the second wave or something, we were doing 10 lakh tests per day, 1 million a day, RT-PCR tests. So certainly there is a capacity, there is a tremendous potential to scale up things very quickly. But our preparedness will largely depend upon how transparent way and how fast we share the information, which is where we have to learn. Lots of genome sequencing was done, but the sequences are waiting in the computer in the lab for several weeks, sometimes months before they are put in the public domain. And unless you put the information in public domain, it is waste. It's like jungle mein fool. Nobody saw. So what is there is no point in that. You have sequenced and kept in your computer has no value. So we have to learn how to share information. A system has to also learn that knowing the truth is more useful. It may be a shocker at that moment, but knowing that there is a problem, you can do something. But if you don't know the problem, don't want to see the problem, then you will have more difficulties. So we have to make several improvements, communication to tell people what to, what not to do, how to stop the virus, how not to, why wearing the mask help, and show the data, show the results, so that people get convinced. At the same time, we should indigenize, don't depend on import or the cheap supply from China, because when the tough time comes, you might not get it at the moment, at that moment. So therefore, every essential item should be, we should be Atmanirbhar in that, which is the correct policy government is adopting, and very, very important. And we are quite well, as I say, as prepared. But having capacity, having potential is not enough. Deploying it appropriately, putting right policies in place, sharing the information is important, and that's dynamic thing. So you may have a very good bat and ball, but you should know how to play the cricket to win the match. So capacity is not enough. I think the ability to decide and implement, take correct decisions are important, and that can happen only when the experts are taken. I mean, they're trusted, and the data is available to people to analyze and give their precious suggestions and inputs.
Sir, there is one more question. I think it's the last question. Is it possible to predict the future mutations and their impact on viral replication using simulations?
No, it is not possible, because future mutation is a random event that doesn't happen by any design. It's just error of replication, and in this virus there is no correction mechanism, no editing mechanism, so errors are more frequent. Since virus in each individual virus will have make millions and millions and millions of particles, so even if 99% of them are mutated badly and they are not of any use for the virus to propagate, it doesn't matter, because a small fraction is already in hundreds of millions in number, so it can infect. So that's why virus uses the number as its strength. But replication, I mean, error replication is totally random. I mean, error during replication is totally random. It can't be predicted or simulated. You can simulate that if that mutation happens, what might be the property that you may get to see in the virus, but you cannot predict which mutation will come, because that's totally random process. After that, natural selection will operate, and whatever helps the virus spread will be more successful and be seen more. And whether it's by more infectivity or it's by evading the immune response, but often milder symptom is also advantageous for the virus, because unknowingly we are spreading. If we fall very sick, then we will not be that mobile and all, and virus will suffer, because it will not be able to reach many people. So that helps, and that is how gradually the virus has become very infectious but mild, and our body immune systems and vaccines help, and then they reach toward the endemic point, and maybe that is where we are now heading.
Okay, I think no more questions are there. It was really very encouraging, wonderful, and informative talk, sir. That I like the approach, the way we have done. So, Akhilesh,
Thank you very much. Thank you for
uh,
listening, and very nice to have many questions. So people are very active. So thank you so much for
your
organizing
talk
this
was
event.
such a way, sir, introducing question. So here, Anona, can you hear me? Akhilesh?
Yes, sir.
1 second. Okay, thank you, sir. As a vote of thanks, because it's an online mode. So, sir, as a vote of thanks, here we are giving you this virtual memento. And thank you so much, sir.
Thank
Actually,
you.
yeah, it will be nice we can get in offline mode, but it was really wonderful talk, sir. Thank you so much, sir, for giving us piece
Thank you
of
so much.
time.
Nice of you. Thank you.
Okay, thank you, sir. Now we can start the next talk. So we are delighted to have Professor Krishna Misra ma'am with us. She is one of the leading scientists in India. She is currently honorary professor at IIIT Allahabad since 2003. She holds honorary position at CBMR, SGPGI Lucknow since 2006, and she is associated with National Academy of Science Allahabad since 2015. She also holds the position of Chief Advisor at India Pesticides Limited, Lucknow till 2006. Professor Misra also served as the chairperson of STEM program of DST, Government of India. She also acts as a member of Advisory Committee of Biotech Park, Lucknow. Professor Mishra worked at Allahabad University since 1965 as a postdoctoral fellow, followed by lecturer, reader, professor, head of the Department of Biochemistry, and also as a coordinator for Center for Biotechnology. She completed her BSc and MSc from Agra University in the year of 1956 and 1958 respectively. She had obtained her PhD in chemistry of natural products from Delhi University in 1964. She has published 222 papers in peer-reviewed national and international journals of high repute and has delivered more than 100 presentations. She has been awarded several research projects by CSIR, UGC, DST, DBT, and UPCST. She received 3 joint research projects with ISIS Pharmaceutical USA, Michigan University USA, and H Pharma USA, and 1 industrial project from DST, Government of India. Dr. Misra has filed several patents and granted a number of patents. She has published many books and written around 12 book chapters. She supervised 50 PhD on Doctor of Philosophy and DPhil students and a large number of MSc and MTech students. Most of them are on key position in India and abroad. She had been on task force of DBT, Government of India, and become expert in several selection boards in universities and research institutes across India. Professor Misra had been General Secretary for NACI for 5 years. She visited various countries including UK, Japan, USA, Singapore, Philippines for several times for delivering different lectures, attending workshops, and as UNESCO Fellow. With this brief introduction, I would kindly request Professor Krishna Misra ma'am to deliver his talk on research methodology as how to take up research in right perspective. Over to you, ma'am.
Okay, thank you so much, Dr. Samantha. Am I audible?
Yes, ma'am.
Okay. So, good evening to everyone. And I'm on the very onset, I'm thankful to Dr. Nidhi Mishra and Dr. Sindhu Samantha for inviting me to speak to on this occasion. Well, I would like to say that it is a very proud moment for me. That preceding lecture was by Dr. Rakesh Mishra, who had been my worthy student. So I think there can't be a more bright moment for a teacher to have such an illustrious student. So he gave a very informative lecture. All of you must have enjoyed. I'm going to talk about research methodology. Let me first share first. So I'm going to tell all of you a very basic thing, because I think all participants here must be involved in research in some way or the other. So I thought it is best for me to speak to you that research should always be taken in right perspective. So let's see uh, uh, what is, first of all, what is the meaning of research? Research, as the word indicates, that is searching something which is known, which is present, I should say. And research actually is very systematic, controlled, and this is based on experiments. And that is okay. 1 minute. Why is it disturbing? Okay. Now, what is exactly research? If we see as such, there are three important things uh, world over. One is space, energy, and materials. So, in research, any of these is involved, and you can say that research is first-hand information. Carrying out a laboratory experiment, either you conduct a laboratory experiment, or you do some survey work, or analyze some statistical data. So sometimes, you write reviews. A review means that you are examining what other researchers have already done. So that is present in journals, books, and scientific debates. You can get any information. But then what you do in reviewing, because first of all, anybody
Sorry, ma'am,
starting.
sorry to interrupt. Ma'am, the PPT is not visible, I guess.
Hello?
Ma'am, the PPT is not visible.
PPT is not visible?
Yeah, so I think it's for everyone. Na? PPT is not visible.
Yes,
Yes,
sir.
sir.
PPT is not visible.
Ma'am, can you please?
Uh, well, in that case, can you ask someone to upload?
But I can see the with title, what is meaning of research?
Yes, ma'am. It's looking to some few people, but not all. I don't know why. Can you please, ma'am, close it and can you open it one more time, ma'am, please?
What is the problem? Is it visible now?
No, ma'am.
Nidhi, there is some problem here. I think you can ask someone to. I have sent you in mail my PPT. You, yeah, ask someone to play it. Huh?
Yeah, now it's visible. I guess it is visible to everyone.
Hello.
Ma'am, it's now visible, ma'am.
Yeah, is it visible now?
Yes, yes, yes. You can carry on, ma'am.
Okay. Okay. So I'm sorry for the interruption, and I'm going to speak about in which way exactly the research should be carried out. So, first of all, what is the exact meaning of research? Research is something uh, you have, if you have some concept in your mind, for specifically for scientific research, you have to have some concept in your mind. And that concept, you start with a concept, then you start thinking about, see the literature, what has been done in that specific field, and then it changes to hypothesis. And then you start laboratory testing, or else a survey, or analyze some statistical data you collect, and then you start analyzing that. It may be any of these methods, or it may be that you are reviewing the past researchers' work, and seeing the journals and collecting that, and then in view of your own concept, how critically you can analyze that and give a review. That is exactly what you do in research. So in short, we can say that it is amalgamation of two: the literature search and laboratory research. So research actually allows you to pursue your interests. It should be, the subject should be of your interest, and then you should learn new things. Here you learn to face challenges, because as you have seen from many lectures and many scientists, they speak about challenges. They took some challenge and tried to prove that. So you are developing here your problem solving skills that you sharpen doing research. Next slide, please. Can you change the slide? Yeah. So exactly what is research methodology? Research methodology means it is systematic, theoretical, or practical analysis of the methods applied to field of study. So it comprises theoretical analysis of the body of methods and principles with whatever field, whatever branch of knowledge you are concerned with. So that is research methodology. Next slide. Now, who should do research? Because it's not everybody's field. You should have curiosity to investigate in depth. First thing, you have to be a good organizer, then have patience, because you don't get positive results always. So you have to have a lot of patience. Curiosity is very necessary. Unless you are curious about things, the example of Fleming, that he saw, he was culturing bacteria, and then he left the slide in sink. He went for leave, came back and saw that some of the bacteria were lysed, and saw that that was due to a fungus. He analyzed the fungus, and that was how penicillin was discovered. If penicillin was not discovered, then we wouldn't have had all these antibiotics today. So it is, the curiosity to investigate. They see so many things every day, but then investigate. That should be there. Aptitude for good expression in speech and writing. You have to be a good communicator and a good writer also. You should be able to work in a team. Then hard work and total commitment. That's very essential in research. I think I'll advise the students present here to see on YouTube the latest example of Mr. Sudhanshu Mani, who has made this fastest train running at 180 km/h, and how he managed, how that commitment to work beside all struggles, that is very, very necessary. There are many other examples of such hard work. So by carrying out research in any subject, you are leaving a legacy for the whole world, because science is global. So you don't have any chance of putting any wrong data or any dishonesty or plagiarism. There is no space here in science, because whatever you do will be known to the whole world. So you have to be creative and very open for making discoveries. Grow up as independent and critical thinker. Should be able to sharpen problem-solving analytical skills. You must know that develop skills, develop novel skills, meet people with similar interests, should apply concepts learned in coursework to real life. That is very, very essential. I think this new education policy 2020 has come, and that has I'm very sorry if implemented, that has very good ideas about innovative research, because there should be innovation in research. Otherwise, it won't be of any benefit to the society. Next slide. Now there are 10 best qualifications of a researcher. So you should know the person who should come to do research must have these, at least these qualifications. He or she should be able to take initiative, eager to assume responsibility to improve skill. Then have curiosity. As I said, you have to have curiosity to know things. To whatever you observe, you should be able to analyze properly. Leadership role, always ready to take leadership roles. Then teamwork, because science cannot be done in isolation. It has to be a teamwork. take criticism graciously. If somebody criticizes you, don't feel anything. That is very good, because criticism leads you for better work. So that is your guidepost. Face high pressure at work. Never get discouraged with negative results. Think of new ways to achieve success. Scientific research does not always lead to positive results. You are very likely to get negative results. And if you get negative results, don't feel discouraged, because I know that some people, they are thinking of starting journals only for publishing negative results, because you have done work, but you have not got the results accordingly as you thought it should be. But then that is research. You have to go an extra mile, relentlessly achieve your goal, never settle for mediocrity. Exude confidence. You have to be confident, but never be arrogant. There is difference between confidence and arrogancy. So be honest, report correctly your results, even if not in line with your expectation. That's what is very, very necessary. And you have to be patient, repeat your results with patience. Sustained commitment leads to success. Next slide. Now, research is done through projects, because any innovative research needs money. So it is through projects we do research. And these projects are sponsored by different institutions. There are sponsoring agencies in the country. And there is one principal investigator of the project. His or her responsibility is managing the research and for achieving success in the project. Then there are other people. The principal investigator should select proper people to work in that project. They should be trained properly, and the principal investigator should evaluate the project. Then there has to be a co-coordinator of the study team, and the co-coordinator should make make protocol for work, prepare study materials, conducts reports. So there are different organizations they are sponsoring. Sponsor is responsible for implementing, maintaining quality assurance, because you are using public money in research, and you have to be very careful about this, that ultimately you give something beneficial to the society. So ethics and high quality of research are ensured by committees. There are many committees: internal board reviews, ethics, research committee. So in conducting life science experiments, biosafety also is very, very important. Next slide. So people say that, some people say that research is waste of time and money. But it is, let's see, even if research project is accepted, suppose your research project is accepted and you have appointed right people, and with great commitment you have started working, but is it that you always get the results which you have conceived? May not be. You may not get the results. In most of the time, you get negative results. So here, full commitment, dedication are very, very essential. The main objective is that students must learn. Skill development is the main objective. Take that analytical, conceptual, critical thinking skills to the highest academic level. That's what you are supposed to achieve in doing research. Now, postgraduate students thus prepare themselves for a future job. They are looking for a job, and they must advance their knowledge for the good of society, concentrate on meaningful research, and select topics being appropriate to public benefit. This is very, very essential, that once you select your problem for research, you should always think that how that your results are going to be benefit for the society. So research is never waste of time or money if you carry it out in right perspective. Next, please. Now, as far as technology is concerned, there are three phases. First is the knowledge, basic knowledge of science. Now that leads to invention. Invention is something making new, and then if you acquire knowledge in basic sciences, you can apply it. Now, applied science is developing skill. So that is innovation, something new, something novel. And this together leads to technology. And technology should diffuse. Diffuse means that it should spread. It should be beneficial for the whole society. It should spread throughout the society. People should benefit from your technology. Next. Now, science means solving problem. Technology is transforming the basic knowledge, and innovation means the impact on society. So there is this word innovation, this has been added a decade or two back to scientific technology. And innovation actually is the development of new values through solutions that meet new requirements, inarticulate needs or old customer and market needs in value, adding new ways. I mean, innovation means that whatever invention you do, that should be benefit, that should diffuse into society, like mobile phone. Earlier they used to have that, first discovery of sound, and then telephone was invention. Now mobile is innovation. So this is accomplished through more effective products, processes, services, technologies, ideas that are readily available to markets, governments, and society. That is what research is really meant for. Next. Now here you can understand clearly that innovation, this word, that innovation means what is desirable to users. They should be users, and what you can make with technology and what is viable in the marketplace. So all these three make innovation. Next, please. Now, I'm taking one aspect of science research, that you have to become science communicator, because whatever you do in the lab, you are doing research in a lab, but it is your duty as a scientist to make public aware as to what you are doing, what is being done in the laboratories, scientific laboratories, that people should know. They should be aware of it. So therefore, you have to become a good communicator of science. So this is, so far, specifically in our country, we have not thought about this aspect, but now government is very serious. And government has now become aware of the role of science communicators. And they have started several schemes and projects for this, and this demand is slowly increasing. So for young researchers here, this is to make their future career, they can become good science communicators. So actually, government has made a provision in many different agencies, NGOs, institutes. They are hiring people with good science communication skills. So DST, DBT, ICMR, DRDO, CSIR, all these organizations have started fellowships for science writers. You have to be good writer also, you can become editors of the journals, and you can be popular science writings, so that people, public at large should know what is being done, what is the latest this thing in research. Particularly ladies, for women, there are many programs because they can sit at home and do the job. Online, they can communicate. So this NCCSTC, that National Council for Science and Technology Communication of DST, this has been established to integrate, coordinate, catalyze, and support science communication and popularization. National Institute of Science, this NISCOM, CSIR, has been publishing popular science journals, books, all academies, this Nasi National Academy of Sciences and Bangalore Academy, they are all publishing very good journals, that is for making public aware of the scientific investment. Next. So there is very bright future for science communication in India because several NGOs, they are organizing science communication programs. There is one Indian Science Writers Association, ISWA, that is in head office is in Delhi. That's a professional body which undertakes training courses, lectures, fellowships with other government agencies in promoting science communication. Science-based films, books, including science fiction, that is also becoming very popular. So future prospects of science communication as a career look to be very promising. More young minds need to be attracted to this area, and more courses on science journalism is to be developed in different universities, colleges, to create dedicated science communicators. They are very, very much needed for spread of scientific knowledge. Next. Now, you can communicate science in different ways. There are different ways. The best method is to publish papers in journals, but that is only for the experts in your own field, because only specialists in that field will read your research paper. Review paper also is for specialists only, because review you can write reviews because there it becomes easier for other workers working in that field to get all information in one place. So reviews also are very essential. Then when you start doing research and you reach a level that now your research, you have some positive results to declare, I think the first thing should be to get a copyright, to get this thing on your finding. And that is the best way, is to present your paper in a conference. Or then later on you can publish it in some journal, and you can also write popular this thing in newspapers or science magazines. Then there is project proposals. All these are written in different ways. The language is different, the format is different, and students at this level should learn how to write all this. Project proposals are written in a different way. Then thesis, dissertations, then popular science newspaper articles, oral presentations, posters are made in different ways. Then there are reports, case studies generally in medical profession. They write, if there are very specific things to write about special cases, patients. Then interviews and discussions, computer mediated information. These are different types of scientific writings. So one has to learn the skill of writing also, and then speech, communicating science. Next. What is basic requirement for communication? Because it is global. As I said, the whole message should go to the whole world. There is ABC of research communication: A for accuracy, B for brevity, and C for clarity. So there, you should remember that it should be brief, accurate, it should be coherent, logical, clear, and adaptable to audience. See, the audience is very important for different type of writings. Every article written is by keeping in mind your audience. Because if you are publishing a research paper, your audience will be the specialist people who are either working in your field or they are specialists in your field. Only those people will read your paper. If it is a popular article in a science magazine, then many people, who do not even know that, who are not experts in your field, will read that. So it has to be written in a very different way. The language is different and way to describe is different. So you must learn these skills while you are doing research. Next. Yeah, this is the major components of effective communication: who, why, what, and how. Who are you addressing? Who is your audience? And why? Why is your message important? What is your take-home message? And what is the methodology? How are you giving the information? So who, why, what, and how. This is the message. This is the major components for an effective communication. Okay, next. Audience is most important. Everywhere, whether you are giving an oral presentation, whether you are publishing papers, or whether you are giving a popular article, whatever, but keep audience as utmost important. Effective communication is not only delivering message, but also includes interaction. As we have seen in the previous lecture, the interactive session has been very important. So always ask questions. Who are you addressing? First thing, why is your message important? What is your take-home message? How to best deliver your message and satisfy audience? So popular science writing is equally important for communication, your results in scientific journals. Next. This message determines the medium for formal publication. See, number one, what message do I want to convey? This is for you to think. These questions should come to your mind. What message do I want to convey? What format is most appropriate for my message? Who will be interested? And where should this paper be published? This is one question which every research student faces, that to which journal we should send. So first thing keep in mind is your audience. Who is going to read that? So then only decide, and at what stage your results are. Which message, at that stage, how can you convey your results to the audience? That is most important. Next. Yeah, this is exactly how you should start research. See, you can see at the top, there are question. Question means, that first thing which should come to your brain is concept. What is your concept? Now, there are, in any field, in whichever field you are starting your research, you have to have something important in your brain, in your mind, what you want to work on. If you are interested in something, then you will be really committed and you will take the challenge also. So first thing is question. What is your concept? Then start reading literature. See on your right, you see answers, known or unknown. Whether whatever concept you have, you read literature, then that concept changes to hypothesis. It becomes a hypothesis. Now you have to validate that hypothesis. So you have to find, as a result of reading literature, you find that whether answer to your question is known or unknown. Then if it is unknown, then you find a window in that. Window means that you have to keep in mind many things, whether you have infrastructure to do that research, that much research, and how will you conduct. Then you get the concept of problem. As I said, that concept changes to hypothesis. Then you plan your work, how you are going to do that. That leads to experimental experiment. You can do wet experiments, you can do survey work, you can do statistical analysis, whatever you do. But that leads to data collection. Data collection, then analyze your results, interpretation and conclusion. Then if you get the do analysis, then you will get some results. So you conclude something, and that leads to a new knowledge. Now, at this stage of new knowledge, then there are three alternatives. One is to publish that. or any alternative form. You can go to a conference, you can publish your paper, but then another question, as you solve this, another question may arise in your mind that I can solve this also. So the cycle again starts. But then the third alternative is that you publish a scientific paper. For publishing scientific paper, there is a format known as IMRAD. I for introduction, M for material methods, R for results, D for discussion, and then ultimate conclusion. So that's how these are the main sections of your research publication. Next. Yeah, title of paper is important. That has to be very brief, catchy, and clear. What is the paper about? Then abstract is written at the end, and that is short summary which can stand alone. That means no references, nothing. That should be independent piece. Introduction, known or unknown about the problem. What are your objectives? Material and methods, what work has been done. Then results, what has been found. Discussion may, how you interpret results. Conclusion is probable implications, impact. Acknowledgement, who contributed and how. References, how to find the referred papers. Then supplement, something which you cannot write in the paper, you can give as supplementary material. So that's how this is the summary of main sections of a scientific paper. Next. Now I'm coming to impact factor, because generally, when you decide to publish your work, you think about in which journal you are going to publish. And the first thing comes to your mind, what is the impact factor of the journal? So what is the impact factor? Impact factor, or journal impact factor of any journal is that is the index, that is a scientific index, that is measure of the frequency with which the average article in the journal has been cited in a particular year. That depends on how the papers published in particular journal are cited. Now, this is used to measure the importance of rank of a journal by calculating the times its articles are cited. How many times each article in a journal is cited? That is important. So impact factor is not associated with quality of the journal or what is their reviewing process, peer review process or whatever, but it only reflects the average number of citations of articles published in journals. So impact factor can be calculated after completing the minimum of 3 years of publication. For that reason, journal impact factor cannot be calculated for new journals. Since for 3 years, you cannot calculate. The journal with the highest impact factor is one that published the most commonly cited articles over 2-year period. So 2 years are essential, 3rd year assessment. So after 3 years, the impact factor of a journal is given. Next. Now the impact factor was, I think the previous one. Yeah, it was first given by Eugene Garfield. He was the founder of Institute of Scientific Information, that is known as ISI, a academic publishing service, started as long back as 1956. So relative number of citations an individual article receives is better evaluated that they are known as citation impact. Suppose my paper is cited by 40 people, then 40 is the citation impact. Now, if given in given year may the impact factor of a journal is the average number of citations received per article average published in that journal during 2 preceding years. So actually, it was by Thomson Reuters that they took from this ISI Institute, and they started indexing the journals. Next slide. Next one. Yeah, this ISI and Thomson Reuters, uh, they Thomson Reuters took it from ISI in 1992. And then in 2016, now Thomson Reuters and ISI together, that has been taken over by Clarivate. So Clarivate is the indexing this thing now. Scopus also provides a higher citation count than ISI say, and both in the sciences and in social sciences, humanities, as well as every field. To check if your manuscript is indexed in ISI Web of Science, you can log on to. Once you log on, you can simply search by full journal name, ISS number say, and search result will show you if your target journal is yes, SCI journal. So that's how now it is not Thomson Reuters, but it is Clarivate. Clarivate and Scopus is another, this little bit confusing. Next. The concept is what factor we have to take into consideration. Some high quantity, because this Thomson Reuters, ISI, Thomson Reuters, or now Clarivate, they don't index all journals. So there are very good journals which are not indexed by them. So high quality reputed journals are not forwarded to Thomson Reuters. So other e-journals are having different impact factors, but also are considered. Therefore, this is a bit complex, that all SCI journals are not, you shouldn't say that it should be only SCI journal. Next. Yeah, this is the type of calculation. This is just for the fresh students, that how do you calculate the impact factor of a journal? I've given here an example of Nature. Nature in 2017, the impact factor was 41.577. That is the total number of citations in that year divided by the sum of publications in previous year and previous to that, 2 years publications. So that means that on an average, Nature's paper published in 2015-16 received roughly 42 citations each in 2017. But then the impact factor is given in 2018 because all papers published in 2017 have to be taken into account. So that's how impact factor is calculated. Next. Now I come to another thing, ethics. Ethics is very important in research. You should not fabricate, you should not falsify, misrepresent any of your results. Research involves coordination at global level. So ethical standards are essential for collaborative work. Many ethical norms such as guidelines of authorship, copyright, patenting policies, data sharing policies, confidentiality rules in peer review are designed to protect intellectual property interests while encouraging collaboration. Because research cannot be done in isolation, it has to be collaborative, so there is no place for these falsifications. Researchers want credit for their contributions and do not want to have their ideas stolen. So people are more likely to fund a research project if they can trust the quality and integrity of research. That is what is very, very important. Next. Ethical lapses in research can significantly harm human animal subjects, students, and public at large. A researcher who fabricates data in a clinical trial, he can harm the patients, even kill the patients, or researcher who fails to abide by regulations, guidelines relating to radiation or biological safety, like safety from these viruses or any pathogens, that may jeopardize his health and safety health. So safety measures are extremely important here. Next. Now, principles in ethics are honesty, number one, do not fabricate, objectivity, Strive to avoid bias in experimental design, no bias. Data analysis, interpretation. Peer review may no bias, no. Integrity, keep your promises, agreements. Carefulness, openness, respect for individual intellectual property, copyrights. Do not use unpublished data, methods, results without permission. Give credit where credit is due. Do not plagiarize. Plagiarism is an academic crime. Next. Now, these are some of the principles of ethics: confidentiality, responsible publication, responsible mentoring. Mentors have to be very careful, they should educate, mentor, advise students, promote their welfare, and allow them to make their own decisions. You can help as mentors, as supervisors, you can help students, but you make them do their own work, make their own decisions. Respect to colleagues, respect your colleague, treat them fairly. Non-discrimination, there should not be any discrimination based on sex, caste, ethnicity, or any factor. Competence, maintain, improve your professional competence and expertise through lifelong education and learning. Legality, animal care, if you are using any animal, you have to design your experiments very carefully. Human subject protection. So while conducting research on human subjects, minimize harms and risk, maximize benefits. Respect human dignity, privacy. Patients' data should not be declared, and autonomy. Next. Research misconduct is fabrication of results, falsification, plagiarism. Research misconduct does not include honest errors. Sometimes there are, there is some difference of opinion between two workers. That should not be taken as misconduct. Next. Now, what is plagiarism is another, practice of taking someone else's work or idea, passing them off as your own. Legal dictionary describes it act of appropriating the literary composition of another author, experts, ideas, or passages therefrom, and passing the material off as one's own creation. This should not be done. Next. Wikipedia describes it as wrongful appropriation, purloining and publication of another author's language, thoughts, ideas, expressions, and representation of them as one's own original work. That is plagiarism. Next. Now, there are 10 main forms of plagiarism: submitting someone's work as your own, taking passages from your own previous work without adding citations, rewriting someone's work without properly citing, use quotations but not citing the source, intervening various sources together in the work without citing some but not all passages, meddling together cited, non-cited sections, providing proper citations but fail to change the structure, wording, inaccurately citing the source, relying too heavily on other people's work. All these are under plagiarism. Next. There, it may be wholesale. That means you copy the entire thing. Patchwork, patches may be ho sakta hai. Idea based. Someone's idea, basic idea is there, but you are writing as if it is your original idea. So that's also plagiarism. Sprinkle, occasional failure to cite properly through missing information. There may be errors in punctuation. This is often unintentional bhi ho sakta hai. That can also be unintentional. Next. Intentional is dishonest plagiarism, and it is really very, this is a academic crime. And paraphrasing too close to original, patch writing, involves intentional copying of substantial proportion of others' work without written or unwritten permission, also without acknowledging the source. So intentional deliberate plagiarism is dishonesty, it is academic crime. Next. Unintentional भी होता है, that means negligent plagiarism, which you do ignorantly, carelessly. It mainly arises from one's inadequate knowledge, competency in writing. It also due to careless attitude resulting in non-compliance of standard verification procedures. So in this type of plagiarism, degree of copying is not substantial. Next. So there are some do's and don'ts for research. Research do's are that you take notes in your own words. Indicate clearly in notes where different ideas come from. You can use some some quotations, some abbreviations to indicate that it is your idea or someone else's idea. Copy direct quotations word to word within inverted commas. Note page numbers. Keep track of bibliographic information and the date you retrieved the information. Save draft versions of your writing as you proceed. So these are some of the do's you should remember. Next. Cut and paste material. Don't do that. Patch writing. No. Forget to cite. No. So these are some of the research don'ts. Next. Avoiding tips for avoiding plagiarism. There are there is a 3-word rule. If you copy 3 or more words verbatim, word for word from a source, you must use quotes around these words and immediately reference the source. Use in text citations. Every phrase, sentence, or paragraph that you copy from another source must be enclosed in quotations. Then you must immediately provide a source. So always cite. Next. Essential requirement for students. All students should necessarily be required to submit a signed statement that they are aware of plagiarism policy of the university institute and no part of their work, be it assignment, term paper, project report, thesis or dissertation, anything is not copied in any form and it is their own creation. So every student, I think, should be made to sign this statement. Next. Punishment is plagiarism is something like copying an examination. Depending on severity of crime, the punishment should be fine. You may be fined, you may be rusticated for limited time, or your degree may be withdrawn if it is too serious. Next. Yeah. So carry out research in right perspective. Thank you.
Participants, ma'am, it was really very interesting and informative talk. So the participants, if you have queries, you can write in the chat box or you can speak out your if you have some doubts.
Thank you. Thank you. Oh, thanks everyone. Thank you.
Ma'am, you have presented so well. So, and you've explained everything so nicely. So I think no one.
Can you hear me, ma'am?
Yes, yes, you're audible. You
Okay.
can.
Good evening, ma'am. So nice presentation. Thank you very much.
Thank
I'm
you.
Dr. Rajkumar from JP University, very nearby to Shimla, Himachal Pradesh. So
Okay.
I have one question for you. It's about uh, the mistakes which either hopefully intentionally done by some of the students, maybe for various reasons, such as uh, completing their degrees, especially.
Yeah.
So recently, what we see is that there are very efficient methods that can actually recognize that some portions of the pictures or the pictures as a whole has been taken from some other source or already published papers. So my question to you is that how, uh, as a guide, should be aware or how can we stop the students from doing so?
Yeah, that's a very good challenging thing, And as I think there are many now, there are many software tools for checking plagiarism. So I think the supervisors, the mentors, should first of all tell them the seriousness of the thing. But if they do it, I mean, any article, any thesis, any paper, anything they send should be thoroughly checked through softwares, because there are many softwares now freely available online which can be used for checking plagiarism.
Especially for the text plagiarism, they work well, but how about the pictures which have been fabricated? So uh, I'm not sure, is there any platform which can actually check out the pictures? For example, some gel uh, pictures which have been actually taken from some of the published sources.
A very good question. I think, yeah, there are many examples of this where pictures have been copied, and they have. I have examples to show that they have taken parts of pictures, photographs from different papers, and joined them, fraction. I mean, parts of pictures or this thing, and combined them into one and published. But that also, now I think some of the tools, very good tools have come for checking this also. Even if a fragment of a photo or a chart or anything is copied, that that can be
Yeah,
checked, I think. Yeah.
yeah, exactly. That was my question. And if you kindly have any source of those sort of programs or any information, so
I can
would
give
you
you the
please
list
like
of all these tools, by which you can check
give?
plagiarism.
Yeah, sure. Sure. Thank you, ma'am. That was a nice talk and wonderful presentation. Thank you.
Thank you. Thank you very much.
Ma'am, I have one small query. Suppose someone doing some experiment and there is a one control, and simultaneously maybe you are running 4 different treated samples. Okay. So now maybe these two of the samples, you are basically writing one manuscript where these two of the sample is going for a paper. And another two treated sample is for other samples, maybe you can go for the next paper. So in that case, the control will remain the same. So do you think is it possible to provide, and if you can clearly mention that this control has been used in this paper, so what do you think? Will it come under plagiarism?
It is not exactly, what I understand what you said, that one student does something experimental, this thing on one sample, the other is doing the on another sample, but the methodology and the process is same. Is it?
Yes, ma'am, I'm telling. Suppose I am running one control sample, untreated samples. Along
Yeah.
with this, I am running 4 different treated samples. Okay.
Yeah.
Now, two of the treated sample is going for one paper, and I'm going to publish another paper. These two of the another your treated samples, but the control remain the same. Okay. So what I am doing,
In
maybe
that
first
case,
case.
if it is your own paper,
Yes,
yeah,
I'm telling my own papers and.
yeah, own paper. But
Hmm.
even if you repeat something from, suppose you have published one paper, and you are now publishing another paper, maybe the students are different, but the corresponding author is same. So you are
Okay.
publishing another paper, you have to cite the whatever you have done in the previous paper also, you have to give, you have to cite in this paper,
Okay,
whatever has
that's what
been
I have to
published
clearly
earlier.
mention it.
Yeah,
We have
and
to
then, if the procedure, you are repeating the same procedure, you need not give the whole procedure here, just the
just
reference
cite
to
that
the-
paper. Okay, okay, okay.
that in the previous paper we have published this is only the data for another sample, and that's all. I mean, a short communication. You don't have to
Yes, yes.
repeat.
Okay. Ma'am, someone asked for giving the list for checking plagiarism. So, okay, we can share with you people. Maybe in future when I'm going to share the recording as well.
I have a doubt. Sorry to interrupt.
Yeah,
Ma'am,
sure.
I have a doubt.
Yeah.
Ma'am, can you please explain me the difference between H index and I-10 index, which we are seeing on Google Scholar these days?
What? I didn't hear that. Can you
Ma'am, the
repeat?
difference between H index and I-10 index.
Which index?
Index and
Ma'am, H index and I index.
No, H index is for authors,
citation number of citation, but
I mean, mostly all these indexes are based on citations. So I think the H index is for authors, or that is, how many times, if your each paper has been cited, and with total number of, divided by total number of publications. So H index also can be calculated for any author.
Any other queries? Participants, if any other queries, please write in the chat box. Ma'am, I think no more queries. I think we
Okay.
have clarified their
Thank you.
doubts. So
Thank you.
thank you so much,
Thank
ma'am.
everyone.
It's a wonderful and informative talk. So I think we'll share the recording as per the ICT norms with all the participants when it's get over. So now with this, I would like to give as a vote of thanks for this wonderful talk, this virtual memento to Professor Krishna Mishra, ma'am. Thank you, ma'am, for this, for giving this precious time and sharing your thought and knowledge with all the participants.
Thank you. Thank you so much. Thank you.
Oh, thank you, ma'am. So participant, do you have any other general queries irrespective of these talks? Any other queries, please? So if not, then I think we can conclude today. And I request you all to please be there and tomorrow at sharply at 6:45 p.m. preferably, so that we can start the presentation sharply at 7 p.m. Okay. Okay then. Thank you. Good night to all of you.